Welcome. I'm Kaitlyn, and this is Kaitlyn and Nhi's knockoff crash course. Today we're going to learn about the ins and outs of our DNA. The three parts of the DNA structure are a phosphate group, a 5-carbon sugar also known as deoxyribose, and one of the 4 nitrogenous bases, which are adenine, thymine, cytosine, and guanine. I'm sure most of you have seen what DNA looks like before, whether it's been on TV or in a science class before now, and seen that it looks sort of like a twisted ladder, and that is called a double helix. And while it doesn't seem very important to know what shape it comes in, it's actually vital to how the component parts are bonded together in the X-shaped pattern that shows how strands in the DNA are twisted around each other like coils of a spring, with 2 strands in the structure. The strands are also considered anti-parallel, meaning that they run in opposite directions, making it possible for the nitrogenous bases in the center of the structure to be able to create a hydrogen bond with deoxyribose. These bonds are very important because they are strong enough to hold the DNA strands together, but weak enough that they can break and allow the DNA to replicate. Another type of bond, called a covalent bond, holds together a phosphate group and deoxyribose, but also the deoxyribose to the nitrogenous base. A scientist named Chargaff discovered a very simple concept that would later lead to an important understanding of base pairing. Chargaff created a rule due to his discovery that the concentration of adenine and thymine and cytosine and guanine are almost equal in any sample of DNA. DNA can only form between adenine and thymine, and then cytosine and guanine, which is what enables DNA to be replicated using only one strand, as only one base could possibly pair with the original, ensuring that the new strand will always be complementary to the original. The rules of base pairing are also used to be able to understand how a cell divides. Before a cell can divide, it must replicate its DNA in a process of replication. This process ensures that each resulting cell will have a complete set of DNA molecules. The first step of replication is when the 2 strands of the double helix separate, thanks to an enzyme known as helicase, forming replication forks. The main enzyme involved in DNA replication is DNA polymerase, and its job is to add and join individual nucleotides to form the new strand of DNA. Then, as each new strand begins to form, their new bases are added following the rules of base pairing. The new DNA strands that are forming are complementary to the original copy, and this leads to the new DNA molecule being identical to the original. Another important molecule of genetic information is ribonucleic acid, also known as RNA. RNA takes the information created by the DNA and converts it to make proteins. While similar, the DNA and RNA have major differences that allow them to do their jobs. The differences between DNA and RNA are: RNA has ribose, while DNA has deoxyribose. RNA is single-stranded, and RNA has uracil in the place of thymine. Having uracil instead enables the RNA to leave the nucleus, since thymine would not allow DNA to do that. RNA consists of long chains of nucleotides, and there are 3 types of RNA. These are mRNA, which stands for messenger RNA, that carries copies of instructions for the assembly of amino acids into proteins from the DNA to the rest of the cell. rRNA, which stands for ribosomal RNA, that combines with the proteins to form ribosomes. And tRNA, which stands for transfer RNA, carrying amino acids to a ribosome during the protein synthesis. These 3 subcategories of RNA are specialized to carry out specific tasks in protein synthesis. In transcription, segments of DNA serve as templates to produce complementary RNA molecules. Transcription uses an enzyme called RNA polymerase to bind and separate the growing chain of RNA nucleotides. RNA polymerase has a similar function to DNA polymerase, but of course it's for assembling new RNA strands instead. One strand of DNA nucleotides are assembled into complementary RNA strand. A specific region of the DNA where RNA polymerase can bind and begin transcription is called a promoter. When certain proteins are needed to be made, not all of DNA is required. Introns are the sequence of DNA that aren't required for coding and remain in the nucleus. Exons are the DNA sequences that are needed to code the protein. They're spliced together to form the final mRNA. mRNA is transcribed in the nucleus and then enters the cytoplasm and attaches to a ribosome for translation. The genetic code of the mRNA is translated into what proteins is needed through codons. Codons are 3 consecutive bases in a DNA sequence that specify a single amino acid that is going to be added to the polypeptide chain. There are codons that signal to stop and start building proteins. Examples of stop codons are UAA, UAG, and UGA, and the only start codon being AUG, which is also called methionine. Translation begins when a ribosome attaches to an mRNA molecule in the cytoplasm, and as the ribosome begins to read the mRNA codons, it directs tRNA to bring the specific amino acid into the ribosome. The ribosome then attaches each amino acid to the growing chain, with each tRNA molecule carrying just one kind of amino acid. A group of 3 unpaired bases on a tRNA molecule that are complementary to the 3 bases of a codon of mRNA called anticodons reads and takes which codons are needed. The ribosome helps form a peptide bond between the first and second amino acids, while the bond holding the first tRNA molecule to its amino acid is simultaneously broken. The tRNA is then moved into the 3rd binding site, where it exits the ribosome. The ribosome then moves to the 3rd codon, where tRNA brings the amino acid specified by the 3rd codon. The chain continues to grow until a stop codon is reached, and the newly formed polypeptide and mRNA molecules are released. You can't subscribe or like this video because we only did this for you, Miss Ray. So you all just witnessed both the birth and the death of Kaitlyn and Nhi's knockoff crash course.